Okay. Welcome, guys. Yes, please. Oh, yeah. I'll do that. I always record everything, you know. All right. So what we'll talk about today is networks. So how do we use the networks that we learned about in the regulatory genomics session? So and sort of understand them systematically. What are some algorithms that we can use once we have these networks built? So on one hand, we're going to be building on the lecture on regulatory motifs from Tuesday, but we will also look at additional ways of discovering networks and then algorithms for studying networks. And then on Thursday next week, we'll talk about a different class of networks, namely machine learning networks. So basically deep learning and neural networks. So today we're going to be talking about network algorithms and the probabilistic interpretation of these networks and some foundational linear algebra techniques that are very broadly useful. So what are these specifically? First, let's introduce networks. What are the different types of networks? Regulatory networks, metabolic networks, signaling networks, interaction networks, functional networks, and then look at both the probabilistic interpretation and the algebraic interpretation. We're going to talk about measures of network centrality, basically both local measures of degree, betweenness, closeness, as well as global metrics, eigenvector centrality, PageRank, etc. Then we're going to look at the algebraic interpretation and also geometric interpretation of these networks. How do we deal with graphs using linear algebra techniques like eigenvalue decomposition, singular value decomposition, low rank approximations, and what does that mean in the context of networks? Then we're going to look at applications of SVD specifically for principal component analysis and ways of regularizing and sparsifying our data sets to basically find the most relevant features and the most informative decomposition. Then we're going to talk about network communities and modules. So algorithm for discovering, for predicting the function of unannotated proteins by association and co-localization in these networks with proteins that we know, the function of. And also, how do we infer cliques? How do we infer modules based on density? And how do we cluster graphs using these decomposition approaches that we talked about, and specifically taking the eigenvalues of the Laplacian matrix of a graph actually gives you, some theoretically very nice partitions. And then we're going to talk about kernels of diffusion and deconvolution of these spread out information by sort of bringing the information back together. Everybody excited about today? Awesome. So, where are networks in biology? It's a trick question because the question should maybe be, where are networks not in biology? Because they're everywhere. They're starting with the genome, about the regulatory networks that we talked about on Tuesday. So there are cis regulatory elements. These are the control regions of the genome that connect to the genes. And these are networks at the cis level, at the local level. There are trans networks that are proteins binding these cis regulatory elements. There are RNA networks of co-expression information between different classes of RNAs. There are networks of co-activity of how the epigenome is looped in 3 dimensions. We talked about that at the end of the second epigenomics lecture, about how we can infer the 3D connectivity patterns of the whole genome. These are networks of chromatin. And of course, at the protein level, there are many, many different types of networks. There are transcription factor networks, as I mentioned. There are signaling networks that do not directly contact the DNA, but bring signals from outside the cell, inside the cell, or from outside the nucleus, inside the nucleus. And there are also metabolic networks that basically guide the transformation of enzymes, so by enzymes of particular metabolites into other metabolites, okay? So many different types of networks, and all these networks are actually interconnected with each other. So metabolic networks that I mentioned just now are guided by the proteins that are the edges at every one of these links that transform these metabolites into other metabolites. There are protein and signaling networks that communicate with both pre and post-transcriptional regulation, and all the way down at the cis regulatory level. So basically you can think of this as layers of information on top of the genome. You basically have the genome, the epigenome, the transcriptome, the proteome, the metabolome, and so on and so forth, okay, at all of these different levels. Everybody with me so far? So basically, these networks are really pervasive, and there's many different types of networks. So it's important to define the types of networks that we have at each of those levels to be much more specific. So at the lowest level, closest to the DNA, we have these regulatory networks. And basically, there are both cis links between gene regulatory elements and their target genes. So some of those are guided by three-dimensional conformation. There are trans links between the regulators and the elements that they bind, okay? So we spoke about both types of links in the last couple of lectures. And these are directed networks because a protein might be an activator or a repressor of a particular gene or of a particular other protein. They are signed because a transcription factor regulates a gene, but that gene doesn't regulate the transcription factor. So they're both directed and signed. It can be a positive or negative regulation. And it's a weighted regulation, which basically says A controls B, controls C maybe 3 times more than B controls C, okay? So directed basically means I know who the regulator is and I know who the target is. Signed means I know if it's a positive or a negative regulatory interaction. And then weighted means I know the strength. Who's 100% with me so far? Awesome. So for metabolic networks, it's a little funny. The proteins used to be the nodes. Now the proteins are the edges. So the metabolites are the nodes, and the metabolic reactions themselves are carried out by enzymes. So knowing how much of an enzyme is there will basically tell me how much of that edge there is. Who's with me so far? Awesome. So they're basically usually undirected networks because you know what enzyme catalyzes you know, a particular reaction, and sometimes the same enzyme might be working both directions, or when different enzymes are catalyzing different steps or different directions, you have a directed network. And this is weighted because it tells you how much of an effect it has, okay? So these are either undirected or bidirected because this metabolite might sort of lead to this metabolite, but not the other way around. Or most of the time, there are symmetric reactions with different enzymes guiding those. Sounds good? Then signaling networks are basically not quite directly touching the DNA, but they're usually upstream of these transcription factors. How does a transcription factor know when to turn on? How does GAL4 know? Well, there are other proteins like GAL8 and GAL2 that are basically guiding the transport of galactose into the cell and the signal that this galactose is in the environment. And basically, those are very often signaling proteins that basically receive signals from receptors. They might be sending that signal informationally through phosphorylation of the target substrate protein, the target protein, and then eventually leading to, for example, the activation of the transcription factor or its localization inside the nucleus or outside in the cytoplasm and so on and so forth. Okay. And then these are both directed because, again, the order of phosphorylation events is directed, and usually they're unweighted because we frankly don't know the weight. There are protein interaction networks, and this is a lot of sort of what people think about when they say, oh, there's a gene network. They basically think of a protein network. These are networks of the protein product. Basically, the genes themselves are sitting on the DNA, but the proteins are the products of those genes. So basically, a gene is first transcribed and then translated to make a protein. And these proteins themselves are now interacting with this whole other layer. And these are undirected and usually unweighted networks. And they basically say that these two proteins can basically touch each other. They have domains that are sort of compatible. Everybody with me so far? And then lastly, these networks, the last class of networks are co-expression networks or functional networks or co-something networks. These are not physically instantiated in the genome. So basically all of those, there is a physical thing that happens. But for those networks, there's no physical thing. Instead, what you have is a functional relationship that basically says that these two genes are co-expressed. Yes, there may be a physical interpretation of that where this particular regulator binds both of them and then turns both of these genes on, and then the corresponding proteins are co-expressed. But it could also be that this one, governs that one, or that one governs this one, or that they both independently respond to some environmental stimulus. Okay, so these networks don't necessarily have a physical basis, and they're also undirected and weighted because the weight basically tells you the strength of the correlation between them. Sounds good? And there's a lot of information exchange, as I mentioned, where at the core, you basically have these cis-regulatory networks like we talked about on Tuesday, and then you basically transcribe proteins from these networks. These proteins are interacting, they form transcription factor complexes, which then feed back to regulate these genes, or they might actually transcribe enzymes, which are then sort of sending signals inside the cell or outside the cell, and these signals will eventually activate additional transcription factors and so on and so forth. So this is not well worked out yet. So basically, a lot of people are focusing on individual networks, but very rarely have I seen models that try to sort of model the full interaction between all these different types of networks. And today, we'll make the same kind of simplification. We'll develop methods that will apply in each of these networks in isolation, but we won't quite get into sort of how these networks talking with each other. Everybody with me so far? All right. So then, what are the different challenges in understanding the networks of biology? The first one is finding the building blocks, basically finding the elements, finding the regulators, finding the regulatory motifs, finding the target genes. And we talked about some techniques for doing that in the regulatory genomics lecture on Tuesday. The second one is using networks to predict cellular activity. So you'd like to predict, for example, the expression level of a gene by observing something about the network, or sort of something about the properties of the regulatory network, and then you want to predict how it will perform in a new condition. For example, when you subject a patient to a drug, you'd like to actually have a predictive model that says how will this network respond to a particular perturbation. Or you may have genes that are well annotated, interacting or being co-expressed or being co-regulated with proteins of unknown function. So what you'd like to do is predict the function of these proteins. So that's sort of a second type of prediction challenge. The third one is inferring these networks in the first place. So you have your gene expression network, your gene expression matrix that we talked about in lecture, 6, that basically tells you how is every gene expressed in a bunch of different conditions. You'd like to be able to use these activity patterns of your genes to infer either the structure of your network, namely which regulators are binding what genes, for example, or what genes are co-expressed and so on and so forth. Or you'd like to actually predict the specific function. So you may be able to say, well, this output variable is a function of an input variable, or something as a combination of these two other things. Okay? So there's a lot of challenges associated with both of these aspects, both predicting the structure of the network and also functional predictions from that. And then the last one is analyzing the structure of the network so that we can infer particular properties about these networks. So for example, you'd like to figure out what are the most important proteins. And very often we study hubs from the degree distribution of the connectivity patterns of all the proteins to say, well, this protein appears to be very central in this network or very densely connected. And we're going to look at different ways of studying those. Sorry. And another thing is that you may want to recognize network motifs. So these are patterns of connectivity that are recurrent in your network, or to find functional modules. You'd like to maybe partition your network to say, well, A, B, and X appear to be working together, but these guys are not, or something like that, or sort of maybe I should cut it here, maybe I should cut it there. Okay, so we're going to look also at methods for finding modules within networks. Any questions so far? Who's with me? Yay. All right, so we're going to talk about probabilistic networks, and on Thursday next week, we're going to talk about networks as graphical models, where basically every node represents a variable and every edge represents dependencies between those variables. So you could basically model the joint probability distribution of a large number of variables using graphs. Why is that useful? Because it basically very explicitly models the dependencies of those variables in a much more explicit way, in a much more intuitive way. So for example, you could have Bayesian networks that are directed or Markov random fields that are undirected, and you may be able to link these variables in this particular fashion. That seems like a nice flow of information from S1 to S3. And what it tells you probabilistically is that if I have observed S2, S1 becomes independent of S3. They're conditionally independent. Why? Because if I don't observe S2, then observing something about this variable here will change my probabilistic inference about the state of that variable there, and therefore change the state, my probabilistic expectation for what the state of that variable might be. Raise your hands if you're with me. Awesome. So that's a type of information flow. It basically says that if everything's unknown about S3, for example, and I learn something about S1, that information flows through this network along these edges. But now if I have fully observed S2, I know the value of each of these variables. I have observed them. It doesn't matter what kind of observations I make in S1, because I have certainty about what S2 is. So that means that condition upon observing S2, S1 changes in my probabilistic estimates for the values of each of these variables has absolutely no influence on S3. Raise your hands if you're with me. Awesome. Any questions so far? Who feels like they're learning stuff? Yes? Good. All right. So we already talked about the difference between physical networks and relevance networks. In physical networks, the edges represent some kind of physical interaction between the nodes. For example, a binding or a transcription activity, or protein-protein interaction, and so on and so forth. Whereas relevance networks instead are more abstract. They don't have a physical instantiation. They might represent a similarity of two nodes. And even though you can figure out the pairwise similarity of all 20,000 genes versus all 20,000 genes, it doesn't necessarily tell you about the physical instantiation of these similarities. So what we're going to do today is represent networks as graphs. So as a mathematical object that allows us to compute on these networks. So we're going to look at weighted graphs or multigraphs or digraphs or simple graphs. So weighted graphs are graphs that have a weight associated to every edge. And these are generally positive weights. Okay, they basically tell you the strength of the interaction. For a multigraph, you basically may have multiple edges between two nodes. For example, if there are many ways of catalyzing a metabolic reaction, you may have different paths that are both heading from B to E, maybe at different rates or different costs and different conditions. A directed graph or a digraph has arrows on those edges. Okay, these guys are undirected, but you can have versions of those that actually have directionality. This is, you may have multiple arrows pointing in the same direction or arrows pointing in opposite directions. And simple graphs have neither multiple edges nor self-loops. Okay? And then we can represent these in a graphical linear algebra sort of matrix-like form. So a matrix representation of a network can be either a binary adjacency matrix that basically simply tells you is A connected with B no matter what the weight might be, or it might be a real valued matrix when you have a weighted network where the edges actually represent the strength. And if you have an undirected graph, it's a symmetric network. And if you have a directed graph, it's an asymmetric network. Who's 100% with me so far? Awesome. And of course, I can calculate the connectivity strength of every node in my network. So for example, I can add up the weights outgoing from B or incoming into B, and then figure out both the in degree and the out degree of a network. Okay? Everybody with me? All right, so this is basically the different types of networks in biology and how we can interpret those. So now let's jump into ways of finding important core nodes inside my network. So I could basically say what is very important in my gene set by looking at the connectivity patterns. And the question is, what is more important, this node or that node? This node appears to be very heavily connected, but this node, even though it only has 3 neighbors, appear to be quite central in the way that this whole network communicates with that whole network. And therefore, if I delete this node, yes, maybe the central, the local network here will change a lot, but maybe even though this node seems on the surface to be less important, its impact, the impact of its deletion might actually be far greater in the information flow of this network. Everybody with me? So basically the question is, how important is a particular node or a particular edge to the structural characteristics of the system, or to the information, the dynamic characteristics of the system? So, you know, high connectivity is usually associated in most of biology with importance, but also high, you know, local information flow between them. What mathematically we're going to call high in-betweenness. I know it's not a very pretty word, but you know, this is sitting in between those guys. Okay. So the first measure of centrality is simply the degree, the number of edges that a node has, either incident to it or going into it or going out of it. So the degree centrality is simply the number of edges that I have, and these can be directed, undirected, weighted, in degree, out degree, etc. Okay. So for directed graphs, I have separately the in and the out degree. For weighted graphs, I have weighted degrees. Sounds good? So that's one measure of centrality. We mentioned earlier that betweenness centrality might be important. So how do I measure betweenness centrality? So I can basically count all of the shortest paths in my graph that pass through the node, and then divide by the total number of shortest paths. Okay, so basically I can take the sum betweenness centrality for, you know, some particular node K can be the sum over all sources and the sum over all targets that are not K or not the same as each other of the shortest paths, the number of shortest paths that go from I to J through K, divided by the total number of shortest paths between I and J. Who's 100% with me on this formula? Awesome. So nodes with a high betweenness centrality control the information flow of a network, and the edge betweenness is defined in the same way, basically the number of paths that go through a particular edge versus the total number of paths. Okay. So here the shortest paths are from A to B, A to C, A to D, A to E, and so on and so forth. And all of these paths are, I can actually trace from A to D, I have to go through B, and from A to E, I have to go through B, and so on and so forth. So basically, these are all the shortest paths that actually go through B, and these are all the shortest paths that don't go through B, okay, the ones that are not underlined. So then I can ask, what is my betweenness centrality? And I can basically get a betweenness centrality score of 5 out of 10. Everybody with me so far? Great. So another measure of centrality is closeness centrality, namely how close am I to every other node. So you can basically look at the sum of distances between this node that I care about and all other nodes, summing over all other nodes, okay? And basically I can, you know, I want my closeness to be inverse to the distances. So it's basically the number of nodes divided by the, you know, average distance to each of those nodes, okay? So for example, node B is the most central one in spreading information from it to the other nodes in the network because it is the closest one to all the other ones. So if I want to spread information through this network, I'm going to go to the one node that is the most closely connected to all the other ones, okay? So the closeness centrality is basically defined as such. And, you know, you see that B here happens to have, on average, you know, distance of 1, whereas the other ones have an average distance of 2 or more. Sounds good? All right, and then there are examples where the most central node might be, you know, this one according to degree, or this one according to betweenness, and those measures can actually disagree with each other. Okay, depending on the application, depending on what you're looking for, you may want to use different measures of centrality. Sounds good? Another measure of centrality is the eigenvector centrality. Okay, so you can basically make xi proportional to the average of the centralities of all the neighbors. So instead of looking at each node in isolation, I'm going to basically say, well, if I flow information from all of the neighbors to all of the neighbors, what is the total centrality of all of those nodes? So basically, I can define that by looking at how this matrix flows information through any one vector. And what is this? This is basically a multiplication between my adjacency matrix and any kind of, you know, scalar set of weights on my network. So I can basically take, 1 over some lambda constant of the sum of this application of this matrix onto X, which is basically the spreading of information of X into, through the network. So in matrix notation, we basically can write that, the eigenvector centrality of X is basically that product of this matrix with this vector. And to make the centrality non-negative, we can select the eigenvector corresponding to the principal eigenvalue. Okay. So let's now dive into a little bit more of these sort of linear algebra interpretation of this network. Okay, so the last part was jumping ahead a little bit. Let's now review linear algebra. Okay. So as I mentioned earlier, we basically have this adjacency matrix view of graphs. So I can have any graph here with some number of nodes, and I can basically represent the directed network that has a connection between 1 and 2, that has weight A, as A here, and then 1, 2, 3 that has weight F there. Everybody can see this? So basically my adjacency matrix simply tells me the connectivity between every starting node and every ending node. Okay? So this graph basically can be represented as a matrix, and I can choose some ordering, I can number them, and then I can fill in this matrix, and this is simply the adjacency matrix or the incidence matrix. Okay? And the diagonal entries are simply the weights of the self loops. So here I have a self loop between G and between 5 and itself with weight G, and I can actually represent that along the diagonal. The lower triangular matrix basically tells you what's incident upon something, and then the upper diagonal tells you the opposite. Okay, so for a dense matrix, you may have many more connectivities. For a sparse matrix, you will have fewer connectivities. Why is this interesting? Because I can directly compute upon it. Basically, a matrix is an operation onto some kind of vector space. So if I have a 5-dimensional vector space, I can basically apply a matrix transformation on that vector space and transform every vector into some other spot in that high-dimensional space. Okay? So there's a one-to-one relationship between a graph as an information flow operation and the matrix as a multiplicator operation that carries out exactly that information flow. Okay? So if I start with an assignment of weight 5 here and weight 0 everywhere else, I can basically take a vector that has 5, 0, 0, 0, 0 as my initial setting of weights, and then I can multiply that matrix with that vector. So AX will basically tell me the flow of information from that 5 into the rest of the network. Okay? And that's kind of cool because I can then apply A again, and A again, and again, and again. So basically, my multiplication of any kind of input set of weights by this matrix is a transformation of that vector onto the same dimensional space, which basically has weights that now correspond to that information flow. And what's really cool is that I don't need to know that vector in advance to figure out how the network would behave with 2 hops. I can just take A squared without even knowing the input vector. I can take A to the n to basically figure out the full spread of information through that network over n times. And only at the end, I can basically get my vector and then see what applying this information flow to that vector will lead to in terms of the spread. Who's 100% with me so far? Awesome. Who feels that they've learned something so far? Yay, awesome. All right, so matrix operations are extremely powerful because we can now use all of the tools of linear algebra to basically study our networks. And that's what's really cool. It's not just a handy representation for an adjacency matrix. No, it's an operator that allows us to transform any kind of input weight distribution into an output weight distribution through the network. So A is an operator. It's a transformation. It's a shearing matrix onto this 5-dimensional space. Okay. So every node I has two values, you know, xi and yi. Every node updates its label y using the x value from its neighbors, scaled by the label, etc. Okay. So the graph perspective basically shows this very dense sort of information flow. Okay. So being able to think of graphs as matrices and matrices as operations, we can now start doing all kinds of really cool linear algebra tricks. Okay. For example, we can look at the eigenvectors of a graph. We can look at the principal components of a graph. We can look at the singular values of a graph. We can look at low rank approximations of a graph. I mean, it sounds bogus if you're sort of, you know, you have a road network. You know, you take the network of all roads in Boston. What's the eigenvalue of that? Right? Like if you run into a random Bostonian, even at MIT, and you say, hey, what's the eigenvector of, you know, the city of Boston? Well, most of us, you know, like at least at MIT, they'll tell you, oh, it's this root 2 axis. And the other eigenvector is probably orthogonal. It's the 93 axis. But anyway, you see why this is really cool, right? Because we can actually start extracting meaningful information about networks, biological networks, all these different types of networks that I sort of enumerated, through the same type of computational techniques, whether they're directed, undirected, graphed, signed, etc. You can, you know, symmetric. You can basically apply all this linear algebra methodology. Okay, so let's review very briefly. What's an eigenvector? An eigenvector is a vector which, when transformed through this shearing matrix, becomes simply a linear scaling of itself, a scalar multiplication of itself. Okay, it's a shame I don't have an image for that, but basically, in fact, let me see. There you go. So a matrix M is basically an operation on some space, a multidimensional space. In this particular case, I'm showing 2 dimensions. Okay. So for every vector, which basically gives me some kind of coordinate system. So for the vector 2, 1, which may be, you know, somewhere around here, I basically can shear it through my shearing matrix. And then 2, 1 from here will kind of go somewhere around there. Maybe it'll become 4, 1. Okay. So that's what a matrix does to this space. So in the same way that my network can basically look at information flow through it and transform a 5-dimensional vector into another 5-dimensional vector, I can basically transform this through my network and basically tell me what will be the result of that operation. Okay? So what is shearing? Shearing is basically multiplying each of these, adding them up, multiplying, adding, multiplying, adding. It's a series of linear operations. And I can think of these matrix as carrying out a complex combination of operations, or I can decompose it into simple operations that I can then combine. So what is singular value decomposition? Singular value decomposition is simply saying, what are my principal axis of variation, my principal dimensions, my eigenvectors for some classes of matrices, or my singular vectors for a broader class of matrices, that will then allow me to think of this operation, the shearing operation, which is rather complex, as simply a rotation, a scaling, and another rotation. Okay? And all of those are matrix operations. A scaling is a matrix operation with a diagonal matrix. A rotation is a scaling operation with a unit, you know, with no effect on the magnitude of those vectors. Who's 100% with me so far? Awesome. Good. Who feels that they're learning stuff? Good. Awesome. Okay. So this is basically what eigenvectors are. Eigenvectors are those vectors who, in this particular transformation, only become scaled versions of themselves. Can you point to a vector in this picture that is an eigenvector for that matrix transformation? Raise your hands. I have one answer. Good. A ton of answers. Can you yell it out? the red one, right? So the red one does not change direction. Okay, so the red one is actually an eigenvector of that matrix. And pretty much any kind of scaling of the red vector is an eigenvector of that matrix. It is basically those vectors whose direction doesn't change upon applying this crazy weird transformation known as shearing. Okay, everybody with me? Raise your hands if you're with me. Awesome, yay. Okay, so that's what eigenvectors are, and the corresponding eigenvalues is whatever scaling that vector had when applying that matrix operation on it. Okay, so eigenvectors for a square matrix are basically those that obey this particular property, and we can basically think of them in geometric terms, or we can think of them much more simply in algebraic terms. Okay, so in geometric terms, it's the shearing of the space. In algebraic terms, it's simply a bunch of addition, multiplications, and basically two multiplications and one addition in a two-dimensional space. Okay, so the question is, how do I find the eigenvectors and corresponding eigenvalues of a matrix? Well, if that is satisfied, that's simply the same thing as saying that S minus lambda, the scalar, times the identity matrix, that combined operation on V is 0. Sounds good? And it only has a non-zero solution if this is actually 0. And this is an mth order equation. It's a linear equation, which basically tries to find the values lambda that can solve it, and can have at most m solutions, which are basically the roots of the characteristic polynomial. And these roots can be complex, even though S, the matrix itself, is real. Okay, because the product of complex numbers can be, you know, a real number, which is usually basically for a negative number to have the square become a negative number, you need to have complex numbers. Everybody with me so far? This should be reviewed. Okay, so matrix vector multiplication is nothing more than sort of applying this matrix onto this vector. And then if you apply this matrix onto vectors, you know, 1, 2, 3, for example, these are the eigenvalues of that, or the eigenvectors of that matrix, the corresponding eigenvalues are 3, 2, 0. Okay, so for every eigenvector, S acts as a multiple of the identity matrix, but as a different multiple on each of those. And any vector, say 2, 4, 6, can be viewed as a combination of its eigenvectors. Okay, very often we will think of operations that we do on a complex graph as operations, or on a complex set of, you know, values, as operations that we do on sort of combinations of those. For example, you may think, so let's take faces, for example. You know, if I can represent anyone's face as a combination of eigenfaces, I can basically take, you know, a combination of, you know, sort of, you know, someone with a big nose, and someone with wide ears, and someone with, you know, I don't know, a more, you know, round face or square face, etc. And I can basically represent any of you guys as sort of some combination of scaling of these eigenvectors. It's the same thing in biology, basically. If you have a complex response of, you know, of a biological circuit, you could basically break it up into, hey, what goes on in the, I don't know, proteasome pathway, and what goes on in the mitochondrial membrane proteins, and so on and so forth. So you can basically break up the network into its components and then apply whatever operation onto the components. Okay, so it's the same thing here. You can basically think of 2, 4, 6 as basically 2 times the first eigenvector, 4 times the second eigenvector, and 6 times the third eigenvector. Sounds good? All right, so matrix vector multiplication, such as Sx, can be written in terms of the eigenvalues, eigenvectors, which is basically expanded out here. And the cool thing with thinking of this operation as applied to the eigenvectors is that instead of having to do matrix multiplication, I just do scalar multiplication. And that basically means that I'm just adding up a bunch of scalars. Sounds good? So even though X is an arbitrary vector, the action of this transformative matrix on the vector X is determined by the eigenvalues and eigenvectors. So the observation is that the effect of small eigenvalues is actually small, because the smaller the eigenvalue, the smaller of that, you know, that effect will be. Okay. So for symmetric matrices, eigenvectors for distinct eigenvalues are orthogonal. to each other. So remember when I was giving the example of the whole circuitry of Boston, you basically have root 2 and it's kind of orthogonal to 93 and so on and so forth. So basically it's a planar graph. These are basically the dimensions that we're usually going to be thinking about are going to be orthogonal to each other. And going back to our little intuition, these are usually orthogonal eigenvectors of this, space. So basically with those, I can represent pretty much anything using some kind of scalar combination of those. And if they are like this, that basically means that there's some common information flow. Their dot product is non-zero. So that basically means that they're not orthogonal to each other. So I can transform that space to an orthogonal space, for example, by taking the eigenvectors S1 and S2 and the corresponding eigenvalues. Everybody with me here? All right, so great. So these eigenvectors are orthogonal, and then all eigenvalues of a real symmetric matrix are real, and all eigenvalues of a positive semi-definite matrix are non-negative, and these are just some basic properties of these operations. So again, this matrix is real and symmetric, and therefore the eigenvalues are non-negative and they're real, and the eigenvalues are simply solving for this equation of basically taking this matrix minus lambda times the identity matrix, which is simply removing lambda for each of the diagonal items, and then solving for that equation. So if that is 0, then that basically means that 2 minus L squared minus 1 is actually 0. And then the eigenvalues are solutions to this particular equation. And how can this be 0? Well, the only way to be 0 is to be either 1 or 3, because these are the solutions. Because 2 minus 1 squared minus 1 is 0, and 2 minus 3 is minus 1, which squared minus 1 is 0. Everybody with me? So these are the eigenvalues, and the eigenvectors are basically both orthogonal and real. One is pointing in the up right direction, and the other one is pointing to the up left direction, and they're orthogonal to each other. Everybody with me so far? Great. So you basically plug in these values, you solve for the corresponding vector, and that's what you get. So that's where it gets interesting, because now that we have this basic toolkit, we can basically take any kind of square matrix and decompose it into its eigenvalues, eigenvectors, and eigenvalues. Okay, so I can represent S as effectively a diagonal matrix that's 0 everywhere except for its eigenvalues along the diagonal, and that has all of its eigenvectors here and all of its eigenvectors there. Okay, it's actually not the transpose, it's the inverse. It's a little more complicated than that. So basically, the theorem is that there exists an eigen decomposition of any matrix S, which is a square matrix that has at least m independent eigenvectors, because the matrix itself might be defective. It may have a dimensionality that is actually lower than the full dimensionality that is suggested. For example, I could represent a 3-dimensional operation, which in fact is only working on 2D. And therefore, that third dimension, there's simply no vector that goes in that dimension. So I can't ever get there. So I can have a vector here, another vector there, another vector here. Nothing allows me to go up on that dimension. Whereas as soon as I have 2 non-collinear vectors in this dimension, I can sort of expand anything into D, but I can't get to 3D without something that's pointing up. So these matrices are basically known as defective matrices. So if I have a non-defective matrix, then there exists an eigen decomposition that has all of the columns of U as the eigenvectors and all of the diagonal items as the eigenvalues. And this is just the inverse of U, not the transpose. Okay, everybody with me so far? Raise your hands if you're following. Awesome, great. So why is the diagonal decomposition possible? It's just algebra. Basically, if U has eigenvectors as columns, then SU can be written as S times all of these eigenvectors. And then this is nothing more than, because each of them is an eigenvector, S multiplied by the eigenvector is nothing more than a scalar multiplied by that same vector. And these are just lambda 1 through lambda n, these eigenvalues. And then that basically gives me the equivalent of this kind of operation, which is nothing more than the same exact thing, where I have basically taken every eigenvector, and then I just multiplied by the diagonal eigenvalues. And that operation gives me the same exact thing, where every column is an eigenvector. Everybody with me so far? So basically, thus, SU is the same as U lambda, where lambda is the matrix of only diagonal items being eigenvalues. And then I can just multiply both sides with u minus 1, which is the inverse of u. And then I basically have s u minus 1, which is this massive operator, which is the same as this other massive operator, because I've basically added a u minus 1 to this operator. And in the end, I basically can express this operator because these two cancel each other. Again, this is a non-defective matrix, therefore it's invertible as that. With me? Great, so that basically proves that it's possible. So here's an example of diagonal decomposition. You basically can take this matrix, these are the eigenvalues, these are the eigenvectors. I can decompose it by taking this inverse of u. So it's the basically matrix that causes u minus 1 to be 1. And you can see that if I multiply 1/2 with 1, 1, I basically get 1. If I multiply minus 1/2 with 1, 1, I basically have 0 on the upper right. If I multiply these here, I basically get 0 at the bottom because 1/2 minus 1/2 cancel out. And if I multiply that with this guy, I get the bottom right, which is 1. Okay, so this basically means that u minus 1 is the identity matrix. Sounds good? So basically, I can decompose this vector, this matrix here, into this matrix, that matrix, and that matrix. This is a diagonal matrix of eigenvalues. This is the matrix of all of the eigenvectors. And then this is the inverse of this matrix. Sounds good? All right, so I can basically, you know, make sure that everything has unit sizes. So I can basically take the square root, and that basically gives me an orthogonal matrix where every column has unit length and is perpendicular to all the other columns. Okay. So symmetric eigendecomposition is basically that I can scale that, so these matrices are actually symmetric with each other. So now the Q transpose is actually the same as the Q inverse, and this means this is possible all the time when I have a symmetric matrix that's not defective. Okay, so there exists a unique eigendecomposition Q lambda Q transpose, not just Q inverse, where Q is orthogonal and Q minus 1, the inverse of Q is the same as the transpose of Q, and the columns of Q are normalized eigenvectors, and that's sort of where this diagram comes here. Okay, so basically with the symmetric eigendecomposition, I can basically have U and U minus 1 both be the eigenvectors of the matrix S. Is anybody bored out of their mind, or you feel like you're learning stuff? Good. Awesome. Great. All right. So great. So that's for symmetric matrices. What about matrices that are not symmetric, or matrices that are not even squared? Can I still do that? Well, that's where singular value decomposition comes in. Okay. So for n by n matrix A of rank R, there exists a factorization known as the singular value decomposition, which basically has the property that even though A is no longer a square matrix, I now have an n by n matrix. And all of the stuff that I've been talking about here with eigenvalue and eigenvector decomposition just no longer applies, because my matrices are not square, because I basically have all these overhangs. So basically all of these diagonal operations just simply don't work anymore. But I can still take the singular value decomposition. So how is this possible? Basically, I can express A as a matrix n by n that basically has the singular values along the diagonal and some more trailing zeros at the bottom, and that has an n by m matrix of effectively singular values and an n by n matrix of other singular values, which basically together compose back this n by n matrix A. Okay, so this is a much more general operation than eigenvalue and eigenvector decomposition because it applies to non-square matrices. So the columns of U are the orthogonal eigenvectors of AA transpose. Okay, so I'm basically doing a trick to make sure that this works by basically taking A and multiplying it by its transpose, or taking the transpose of A and multiplying it by A. And that basically gives me m by m matrices and n by n matrices, depending on which direction I do the multiplication. Everybody with me so far? So the eigenvalues of this AA transpose are effectively the same as the eigenvalues of A transpose A. And this is effectively a diagonal matrix of all of these, eigenvalues. But because we're now looking at the squares here, these are basically the squares of the eigenvalues. Sounds good? So again, this is the geometric representation of singular value decomposition, which is basically you're taking this complex operation and you're decomposing into a rotation but no scaling, a scaling but no rotation, and then a rotation but no scaling. Okay? And this is basically the dimensions of SVD. You basically have an n by n matrix. You basically decompose that into an n by n matrix and an m by m matrix. And then the singular values are here, which are basically the squares of the eigenvalues of the ATA matrix. Everybody with me? All right. So basically, this is singular value decomposition. Here's an example of basically taking this non-square matrix. And then the SVD is basically this square matrix, that square matrix, and then the diagonal matrix of eigenvalues. Sounds good? Of singular values. All right, why is this exciting? Because I can actually use SVD now to look at a low dimensional approximation of this higher dimensional space. So SVD can be used to compute optimal low rank approximation. That basically means that I'm going to find some matrix A of rank K that minimizes the distance between the original matrix and this new matrix. Okay, so A, K, and X are both m by n matrices, and typically we want the dimensionality to be much, much smaller than the original dimensionality. So to go back to my example of sort of this matrix that has these 3 dimensions, so I could have basically the third vector, the third dimension, being very low variance, very little happens in that third dimension. So a 2-dimensional approximation of this 3-dimensional object would be a fine approximation. And we're going to do the same thing for much higher dimensions. We're basically going to say, instead of looking at 20,000 different dimensions for my gene expression measurement, there aren't 20,000 independent things going on in a human cell. Instead, there are probably only 100 or 1,000 or 2,000. So I can basically reduce the dimensionality by a massive amount. Instead of thinking of every single gene as independent, I'm now going to think of pathways as codependent of each other. Okay? And the way to do that is basically you take the singular value decomposition, and then you just throw away the smallest singular values. It's super simple. You're basically, it's all of the methodology that we worked out already. But in the end, I just zero out the smaller eigenvalue. And that is guaranteed to give me an optimal lower dimensional approximation. Who's with me so far? Awesome. Who feels they've learned some cool stuff today? Yay, awesome, good. So the question, of course, becomes, how many ranks should I keep? How far should I go? And there are some tricks about that are, a little more complex than the scope of this lecture, but you can basically look at the distribution of your eigenvalue sizes, okay? So you can basically choose how many eigenvalues to keep by basically saying, if I have a random matrix where things are basically noisy, where there's no structure, then the distribution of singular values is going to form this semicircle. Okay, this is something that is known as the Wigner semicircle law, so you can look it up. And there's a basically probability distribution of the largest eigenvalue, and you can ask, where is my strongest eigenvalue falling? Okay, you can basically look at these eigenvalues of the non-random matrix and then define some probability for every principal component based on that eigenvalue distribution. And using that, you can basically say, well, there's some values that are way out there and some values that are very small. So those that follow that semicircle, I'm just going to simply ignore. And that allows me to now select the dimensionality of that decomposition. Okay? So what we've talked about is where networks are found in biology, how we can basically represent these networks, both in probabilistic views and algebraic views, and how we can compute network centrality using different ways, both local ways and global ways, and some basic toolkits that we can use to basically compute on these matrices, on these transformation objects. So we figured out how to do eigenvector decomposition for square matrices and singular value decomposition or singular vector decomposition for non-square matrices, and how to do a low rank approximation, which is simply cutting off the smallest eigenvectors, singular vectors, and also this funky Wigner semicircle law that allows you to select how many eigenvalues you should keep. Who feels that they could use a 30-second break? All right, let's do that. Everybody stand up and stretch. It's mandatory. All right, how's everybody feeling? Yeah? I hope you're not maxed out yet. Good. All right, so using this approach of basically looking at these eigenvalues and eigenvectors and the singular values and singular vectors, I can do all kinds of really cool things, including principal component analysis and sparse principal component analysis. So let's cover these briefly, and then we're going to go back to figuring out communities and modules and diffusion. So if I basically have a gene expression measurement of n genes across m different arrays, so for example, I have 20,000 genes in human and 100 different experiments that I've carried out, my number of genes is way, way bigger than my number of experiments. So the question is, in how many different ways did these genes actually move from each other during those experiments? Well, first of all, the experiments themselves do not allow me to capture the full variability, the full expressivity of my 20,000 genes. Chances are that they're going to be highly correlated with each other through these 100 experiments. Moreover, even if I do 1,000,000 experiments, chances are that the human gene regulatory network is only so complex, that there's only so many different things that a human cell can do, rather than 20,000 different possible independent activities. And there's basic biology surrounding that. The first, though, it might be that we simply did not evolve for such huge complexity. The second one is that there's only a limited number of transcription factors, and only a limited number of regulators. So maybe there's only so many different processes I can carry out. And then the third one is simply that this is a very good way to evolve, basically sort of with a modular, compact representation. So the question is, can we take this matrix of m by n and look for a lower dimensional view of it? That instead of looking at 100 different possibly independent experiments, it instead says, well, heat shock and cold shock might just be kind of opposites of each other, okay? And then response to starvation for one amino acid and starvation for another amino acid might kind of elicit the same response, okay? So first of all, on the array side, on the experiment side, I may not have 100 independent experiments. I may have many fewer. So maybe instead of having an n by n matrix, I want to look at a lower rank on the n side that basically says, well, there's truly only 10 different independent experiments. And one of them is the overall shockness, and the other one is the overall starvationness. And then the two together convolve to basically create what seems like a larger number of possible responses. But in fact, it's just the convolution of a smaller number set of responses. Who's with me so far? Awesome. On the gene side, it's the same thing. I may have 20 genes that are all going up and down in the same way. I don't need to represent them as independent dimensions in my data set. I can just collapse them all into, here's the response of this one component of the network, okay? So both on the conditions component and on the genes component, I may be able to sort of reduce that dimensionality. And that's where all the cool stuff that we've been doing comes in. We can basically say, well, if I have n genes and m arrays, I can just look at the singular value decomposition of that. And basically, I can think of m eigenexperiments and how they relate to each other. I can think of n eigengenes and how they relate to each other. And then all of the eigencombinations between. Yes?
This is still choosing some arbitrary k.
Absolutely, absolutely. But the arbitrary k could be a little less arbitrary by looking at the semicircle law. Basically saying, as I remove more and more vectors, dimensions, what is the distribution of the remaining dimensions? And if that distribution looks like a random distribution in the semicircle, I know where I go. If I still have stuff outside the semicircle, I keep using these dimensions. Sounds good? All right, so there's nothing new here. This is exactly all the stuff that we've been doing so far. So we can basically decompose this using this singular value decomposition. So basically we can choose eigenexperiments that are linear combinations of all genes. And then these principal components correspond to some kind of loading vector. And the loadings are simply the coefficients corresponding to those variables in that linear combination. But the challenge with that is that I still have all 20,000 genes. I may have reduced the number of experiments, but all these trailing genes that might not be really responding are somehow stuck with me. And that's where sparse principal component analysis comes in. So the idea of sparse PCA is to reduce the number of explicitly used variables, in our case, genes. So we're going to modify principal component analysis so that the principal components have sparse loadings. Instead of every principal component being defined on the full set of 20,000 genes, I'm going to define it on a reduced set of genes by throwing away genes I might not care about. So we're going to write PCA as an optimization problem with some shrinkage parameters that basically reduce the number of variables. So one popular approach is least absolute shrinkage and selector operation, also known as Lasso, introduced by Tipshirani in 96. And that carries out both variable selection and regularization. It produces sparse models. So the result is you have a modified set of principal components that now have sparse loadings instead of depending on all the genes. So the linear regression problem is the following. You basically have some output variable that is a function of your input variable plus some noise. You have some set of regression coefficients that you scale these input variables by, and you have a multivariate linear model that basically says some function of x is basically a set of scalings on each of the input dimensions. Okay. So the linear regression problem is trying to figure out what is the optimal set of coefficients, which, given some noise, best predict the output variables y from a set of input variables over many different conditions. Sounds good? So there's n observations, p predictors, and the goal is to estimate the coefficients beta. So there's many ways to do that. One way is least squares, where you basically have the squared error from every prediction y, from every prediction, beta xi versus the original goal y. So you basically have some kind of linear space that you're predicting using this linear combination of your x's, and you have some deviation from that, which are the errors, and you're trying to minimize these errors. Raise your hands if you're 100% with me. Great. So basically the goal is to find the betas that minimize that residual sum of squared errors. So the sparse PCA solution with this lasso penalty is basically taking this training set and then calculating the residual sum of squares. And instead of basically exploding the dimensionality to make those betas come really, really close to the solution, it's basically saying, yes, I will care about this, but I will also care about the total sum of all of my betas. Okay, so the more betas I'm using, the more penalty I will pay. So I'm penalizing the number of parameters that I'm using to express this problem. And how am I penalizing that? Well, using just the sum of all of the betas. Okay, so that's a one-dimensional so this is a linear penalty, also known as L1, because it's one-dimensional, it's linear. The pros is that Lasso continuously shrinks the coefficients towards 0, it produces a sparse model, and it allows variable selection. The cons is that the number of variables is limited by the number of observations, And the problem is that we have very few observations in general, and they're much, much smaller than the number of genes. So it will usually select only one of multiple highly correlated variables. And the problem is that the algorithm doesn't care which one will be in the final model. So that's where ridge regularization comes in. So instead of using this linear penalty on the weights, I'm going to use this quadratic penalty on the weights. And the advantage of that is that there's only one solution, and it's a much more stable gradient ascent. The disadvantage is that it's less robust because the square differences lead to the outliers having an overly strong effect. Okay, and one has the advantage of being more robust. The outliers don't matter as much. And then the disadvantage of being a less stable gradient descent, having that multiple solutions. So we have two things that are almost good. Each of them has goods and bads. What are we going to do? I don't know. Let's combine them. So we're going to basically do an elastic network, which is nothing more than some kind of combination of L1 and L2 penalty. Okay. So the objective is to find this minimized beta using both L1 and L2 regularizations. Okay. So the pros is it combines L1 and L2 regression. The limitation of Lasso is removed by ridge, and the limitation of ridge is removed by Lasso, and it selects a group of highly correlated variables where, rather than only one variable for each of them. Okay, everybody with me? So what sparse PCA does is basically constructs this regression framework where PCA can be reconstructed exactly, and it uses Lasso, ridge, or elastic net regression to construct the modified principal components with sparse loadings. Okay. Here's some intuition. Here's how PCA works in this regression framework. Basically have a bunch of training data. And the idea is that every principal component is a linear combination of the p variables, and its loadings can be recovered by regression of the principal components on the p variables. So you basically have all of these loadings and all these coefficients. And then you can basically start setting some of them to 0 using these sum of the beta coefficients or the squares of the beta coefficients. Okay. And there's some simulations, you can make it work, you can find the loadings. This is basically one group, this is another group, and then the rest you can zero out. Okay. Who's with me so far? Yay. All right, so now let's go back to our networks and basically see how all of these techniques can help us learn communities. So what is the thing that we're trying to solve? We're basically, for example, trying to find communities. Why are communities important? Because if I have a bunch of genes in the same community, all of which are involved in, I don't know, liver toxicity, then chances are that the last gene will also be involved in liver toxicity. So this is very helpful in finding co-regulated things or co-associated things or co-expressed things and carrying out something known as guilt by association. If you hang out with a bunch of criminals, chances are I'm going to think you're a criminal. No comments on the current administration. No comments, I said. All right, so the way we can do that is, for example, by doing this iteratively. So, for example, I can, annotate a lot of things and then, find out the remaining things. And every single time I'm predicting one, I could use that as an additional predictor, or I could hide it all the way to the end to have predicted all of them independently. And that's where graph modularity comes in. In order to find these modules of co-associated and co-expressed genes, I want to figure out ways of partitioning my network. Okay, so I can basically have a graph with densely connected subgraphs, and that these densely connected subgraphs are my modules. They're the things that I want to emerge. So the genes in the modules are usually involved in similar functions and are usually co-regulated. And the modules can be identified by partitioning my graph. Everybody with me so far? So we're going to talk about different ways of partitioning the graph, or finding communities, finding clusters. One way is Markov clustering. And that's basically effectively doing a random walk on the graph by starting from a bunch of locations and then seeing how information is diffusing through the network. It's a little more complicated than that because I want to sort of overemphasize the strong connections and de-emphasize the weak connections. So at every iteration, I basically can scale by multiplying one of the vectors of my matrix, and that effectively amplifies the strong weights and it reduces the small weights. Basically, by squaring it, I can effectively make sure that the strong ones get even stronger and the weak ones get even weaker. Everybody gets the intuition? The second one is building these hierarchical communities. And one algorithm is Gervan-Newman, which is one way to build that. And the third one is spectral partitioning, which is basically taking the eigenvalues of the Laplacian matrix. And we're going to look at this one in a little more detail. Okay. Defining communities is hard, it's subjective. There's many different ways of defining communities. You can basically build it around nodes, around groups, around the whole network, or building a hierarchy. And then finding cliques can actually be NP hard. So if I want to find cliques in my network, it is actually NP hard. So algorithmically, with a network of 20,000 nodes, it might actually be super, super, super slow. So you can basically iteratively prune the network to do that and sort of find minimal cliques that way. And you could also look at the overall density and find dense quasi-cliques, which is not NP hard. It's sort of, you can have approximations to that. And you can also build, many different ways of doing this. So as I mentioned earlier, what Markov clustering does is looks at random walks. The problem being, however, that if I have this particular network and I, look at the random walk on it, that basically means that I'm going to take this and multiply it by itself a bunch of times. What that effectively does is that it spreads these initial weights into something that is very diffuse. And that's not great because it has actually eliminated these strongly connected components. So the idea there is that you want to sort of carry out inflation steps, which basically raises individual columns to some non-negative power, and that both strengthens the strong connections and weakens the weak connections. Raise your hands if you're with me on this one. Awesome. And then at every single step, I do my inflation, and then I do my expansion, which basically means multiplying the matrix by itself. Yeah? All right, so the next one is spectral clustering, okay? And that's where the Laplacian of the matrix comes in. So we already talked about the adjacency matrix. That's just the edges, okay? The degree matrix is simply saying for every node, how many other nodes is it connected to? And that number of nodes will be along the diagonal. Raise your hands if you're with me 100% on the degree matrix and on the adjacency matrix. Perfect. So the Laplacian matrix is just the difference between the two. And it will basically have the number, so every single row will sum to 1 because I'm adding the degree and I'm subtracting, all of the off-diagonal items. Everybody with me here? So basically, my diagonal is the degree, and my off-diagonal is spreading that degree to all of the edges that I'm connected with, all of the other nodes that I have edges with. The intuition of the Laplacian is the following. It is basically the gradient of my gradient, or it is the concentration of my gradient. If I have basically a matrix that basically gives me, for every item here, some value, I can look at the derivative of that value. And that derivative will be an arrow pointing to the direction of greatest ascent. And then by looking at the flow of that information on these arrows, the Laplacian matrix effectively allows me to do is calculate the concentration of the gradients. So if all of the arrows are pointing in, this will be a high Laplacian value. And if the arrows are pointing out, this will be a low Laplacian. Okay, so it's kind of like the second derivative, but in matrix notation. Who's with me on the intuition here? Awesome. Another way to think about this is that every single one of those values is going to be connected to another value. So if I'm much higher, the Laplacian matrix is going to be pushing me lower than my neighbors. So it's basically looking at the gradient relative to my neighbors, and it will be positive if my neighbors are lower than me, and it's going to be pulling me down, and the opposite for the other direction. Raise your hands if you're with me so far. Awesome. So the Laplacian is super easy to define and kind of intuitive in some ways. But here's what really makes it extremely powerful. If I have basically the Laplacian matrix for this, again, on the diagonals I have the degree, and the off-diagonals tell me where this degree is spread to, and I can effectively take the eigen decomposition of my Laplacian. And what that does is that it will create a 0 eigenvalue for the first one. And the first non-zero eigenvalue, or the second smallest eigenvalue, will basically give me a solution to this equation that basically says that the Laplacian applied on this vector is effectively a scalar of that vector. And the solution, the eigenvector, will be pointing to pluses on one community and minuses on the other community. Okay? So the eigenvector that has the smallest eigenvalue will be effectively partitioning this network based on the values of this x vector that will be positive for one and negative for the other. And you can see here in this example, all I did was take the eigenvectors, and then the eigenvector with the smallest eigenvalue has in fact partitioned the network in two. Raise your hands if you think this is awesomely cool. Awesome, good. So that's basically what spectral partitioning does. It basically, you always find that the smallest eigenvalue is 0, and the second smallest eigenvalue is basically the matrix that characterizes the partition optimally. Okay? That's where we're going to stop today. We looked at networks, we looked at centrality measures, we looked at basic linear algebra, and then sparse PCA and spectral decomposition. Who feels that they've learned some cool stuff today? Yeah? Awesome. Good. Thank you, guys. See you next Thursday. No class on Tuesday because it's Columbus Day, also known as Native American Day.